The overall goal of this procedure is to provide a simple and safe method for the study of bacterial response to ethylene.
This method will answer key questions regarding plant-microbe interactions, such as the role that ethylene plays in bacterial adhesion and colonization of plants. The main advantage of this technique is that it does not require any specialized equipment, and it's much safer than handling gaseous ethylene in the lab.
Although this method can provide insight into how bacterial cellulose production is affected by ethylene, it can also be applied to other plant-associated microorganisms for the in vitro study of how ethylene-mediated processes are involved in plant colonization.
In preparation, make 100 milliliters of growth medium for the Arabidopsis seedlings and make 100 milliliters of bacterial growth medium for the CEPA decomposition. Once the Arabidopsis medium has tempered, divide 40 milliliters out to a sterile flask and supplement the aliquot with 40 microliters of 10 millimolar ACC. Now load the plate quadrants with 5 milliliters of medium and allow the agar to solidify. Opposing quadrants of all plates have bacterial growth medium and Arabidopsis medium. Use 3 different medium configurations. The CEPA treatment will occur later in the protocol. Prepare each plate configuration in triplicate. Once the agar has cooled, evenly distribute approximately 50 seeds onto each quadrant of Arabidopsis medium. Then, incubate the plates in the dark at 4 degrees Celsius for 3 to 4 days. Later, after incubating the seeds, expose them to fluorescent light for 2 hours. Then, spread 10 microliters